You watching this on YouTube at the same time? What the hell's going on over here? Okay, good morning, everybody. This chapter, I think there's some pretty cool stuff in here, some pretty interesting things, and a couple of stories that I will share with you as we move through and think a little bit about some biotechnology. So we talked last week, we mentioned details about DNA, how we replicate, some ideas about that, and now we want to think about how we might be able to use that for ourselves, or to be able to manipulate things for our own gains, because usually that's what we think about in the first place. It doesn't always have to be that way. So as we start thinking about biotechnology, we start thinking about genetic engineering, which is really what we're going to be focusing on as we move through here. We kind of got to start where it all happened in the first place, which was up at Yellowstone National Park, which seems like kind of an odd place to be thinking about biotechnology, but it's pretty interesting, all right? There are also some interesting things that happened after that because of what has occurred. And you can go to Yellowstone, you can see all sorts of things. Scientists go to Yellowstone, but like me, they keep their hands in their pockets pretty much, right? You can't just wander off and start sampling stuff just because you say I'm a scientist, right? Doesn't work like that anymore, right? Back in the day, it was a little bit different, right? And maybe you wanted a little sample of water from this hot pot, and you could potentially collect that, right? Without any sorts of issues, right? Now it's illegal, right? You can't go and do that stuff. There are definitely scientists that do research up there. It's a huge amount of paperwork in order to give the opportunity just to be able to do that, right? There was a researcher in the biology department for almost 2 decades. He had been on the list to get his permit enabling him to go. He had went with some other researchers and helped them, but could not bring anything back here and actually do science with it because he didn't have the permit, right? So we'll get to a little bit of that in just 1 second as far as why they kind of changed their practices. Biotechnology is big business, right? When we think about genetic engineering, when we think about manipulating things, there's a lot of dollars there, right? And again, we're going back to Yellowstone, all right? So a couple of little tidbits as we think about this, all right? A lot of the biotechnology, a lot of the genetic engineering and stuff that we're going to talk about in this chapter as we progress through here, we think about a lot of microbiology, a lot of bacteria, or manipulating the bacterial organisms, putting something in them that they didn't have before, maybe even our own stuff. And we talked about, you know, extracting DNA. You need to put, if it's a human gene or a eukaryotic gene into a bacterium, you need to use the modified messenger RNA that's removed all of those introns, and then you use reverse transcriptase to make a copy of the DNA that doesn't have all of that extra stuff, right? But in order to get it to amplify, to replicate, to get that DNA to make copies of itself, if you will, in a test tube, requires some specialized enzymes. It requires some special substances that's going to allow this to happen. Back to Yellowstone, right? So there was a microbiologist that I think really pretty recently, if I remember right, passed away. It wasn't too far back. Thomas Brock was his name, and there are all sorts, just like in this course where there's a boatload of different biology textbooks, and deep down, biology is biology, and it really kind of depends on the types of spins those books are putting on the subject, how they try to lure you in and make you think, wow, biology is kind of cool, right? Or maybe it's not so well written and you're like, yep, I knew it. This subject still sucks, right? Hopefully not the case, but there are numerous textbooks, but Thomas Brock's microbiology is a standard, a staple. Most universities, when they're teaching general microbiology, I teach introductory microbiology. We don't use that. There's a lot of details that people that are teaching health professions really probably don't need, right? But if you're thinking microbiology is what I want, almost always you will be holding a copy of his text, right? It's what I used when I went through the microbiology program, right? So he was the scientist in Yellowstone that discovered this type of bacteria, right? Living, I don't know if it was this exact hot pot or not, but something like this, all right, up in Yellowstone. There are all sorts of features. There are lots of different types of microbes that in turn can be thriving in these environments, which really, in some instances, seems kind of impossible, looking at boiling hot water and the capabilities of something to be living in that. That's what fascinated Brock as he was looking at some of the life forms in these hot pots. It kind of was boggling his mind. How is it that these suckers can survive in near boiling hot temperatures, right? Pretty stinking cool, right? We've talked a little bit about some of the extreme bacteria that we now call archaea. And those guys have all sorts of capabilities of thriving in very adverse types of environments, right? Some bacteria also harbor these capabilities of living in really weird types of habitats, right? So he discovers a bacteria called Thermophilus aquaticus, or Thermophilus, right? For high heat lover, right? And living in the water, right? So this organism is capable of surviving, right? He discovers this. There's lots of research that we start thinking about with it, but there's another researcher that is thinking about biotechnology. He's thinking about genetic engineering, right? I mentioned trying to replicate stuff in a test tube, right? We know now how DNA replicates. We know you need the specific nucleotides. You need those ingredients. You need specific enzymes like the, helicases and polymerase for sure. That's going to help to get these things into the mix, right? Lots of protein. When you, apply heat to those proteins, it denatures it. It destroys it. It's like cracking an egg into a hot frying pan, and the albumin, the runny sliminess of the egg, not the yolk, that's all protein. And as soon as it hits the frying pan, it turns white. It's denatured, and it's never going to go back to the slime that it once was inside of that shell, right? So all of these different enzymes, all of these things that are needed for DNA to be able to replicate don't really function outside of the cell, because temperatures are one of the big factors that cause problems, specifically with the enzyme polymerase that's going to bring in the nucleotides and assemble new strands of DNA. Scientists tried and tried to just take these ingredients in a test tube and make this stuff happen, but it wouldn't, right? You couldn't get it to replicate. And if you could get it to replicate, it's like one copy and then it's shot. You're not able to make mass quantities. And that's really what you need if you're thinking molecular biology. If you're thinking some sort of biotech, you need a lot of stuff, right? So Brock discovers this bacterium. Kary Mullis is another microbiologist that is thinking more along the lines of genetics and DNA replication. And he starts thinking about this organism that Brock discovered in a boiling hot pool of water in Yellowstone, and starts to think, how in the hell can this guy be living? What sorts of proteins must it have that enable it to survive at these high temperatures? What's its polymerase, right? That's what he wants to know. What sort of mechanisms does it have that allows it to replicate its DNA in boiling hot water? That could be something we could use, right? We talked about the DNA molecules, and they link together the nucleotides by hydrogen bonding. That's the weakest type of a chemical bond when we think about biology. So weakest type of a bond, a bunch of them together are pretty stinking strong, but they're easy to break and reform, right? Using a little bit of heat, not boiling hot, but pretty close to boiling hot temperatures, you can cause those hydrogen bonds to break, right? And knowing that there's some bacteria that can thrive at near boiling hot temperatures, their polymerase is also functional at these temperatures. So he is able, in a test tube and in a machine that here we see a smaller version, and there are even much smaller ones now. Back in the day, it was something huge like this big box, right? Massive thermocycler, a massive oven that's going to allow things to heat and kind of cool almost instantaneously, right? So Kary Mullis recognizes this enzyme. He's able to isolate it from this Thermus aquaticus bacterium, pulls this enzyme out, and now is able to use it in this machine that he develops, right? This thermocycler, right? The thermocycler allows, like I said, heating and cooling almost instantaneously. Most of them have a little block, an aluminum manifold that little microcentrifuge test tubes go into. And that aluminum block heats and cools very rapidly, right? So as he does this, right, he is able to take a sequence of DNA. You don't need it intact. We're not talking about an intact organism, all of the specific chromosomes. You don't take all of our DNA and replicate it instantaneously. It doesn't happen like that. It's fragments, it's genes or handfuls of genes that get copied repeatedly. And potentially, if we're thinking about, the genome of a bacterium, you would have multiple tubes with bits of each of those that you would be dealing with, all right? So he's able to make these copies, and we call it a polymerase chain reaction, because once we can start this process, as long as you've got the ingredients, as long as you've got all the materials, the nucleotides to build more DNA, the enzymes, the helicase and the polymerase and those that help to twist and untwist the topoisomerases, all of these things don't get used up. They're just helpers to twist and untwist, to zip and unzip, to bring in nucleotides. As long as there are free floating nucleotides, this process just keeps going, right? So as long as you've got all your ingredients, you can make massive amounts of this stuff. Instead of a single copy, we could take millions or billions of copies of that DNA in a handful of hours, right? And that's pretty awesome, right? So Brock discovers it. Mullis takes it a step further, pulling that enzyme out, and in turn receives Nobel Prize, right? So Kary Mullis he's kind of an odd duck a little bit, and I think maybe sometimes, scientists just in general might kind of be described that way in the first place, right? If you win a Nobel Prize, you collect $1 million. That's pretty good amount of money. I think I could be pretty happy with that, right? He in turn, he still does a little bit of science, right? Not so much, but he does. He's got a lab. He also does a lot of lecturing. He'll go and talk to different places. That's apparently really where he's making the bulk of his money, right? And he spends most of his time in sunny Southern California surfing. That's what he likes to do, right? And then occasionally does some other stuff, right? So in biology here, on Thursdays, Thursday afternoon, 4 o'clock, there's a seminar series. They try to get people to come and talk science, and they try to get people that, you know, the bulk of the biology department could be interested in and maybe want to participate, right? So microbiology club, this was years back, they would bring lecturers in, and typically in these circumstances, some stuff has changed a little bit, but usually you find somebody that you want to come and give a lecture. Most people, if you ask them if they will talk for an hour, they're like, sweet, I would love to do that, right? Because people like talking about their stuff, right? So usually when this stuff happens, they're somewhere else, right? They're not living here in Pocatello. We bring them in, fly them in, pay their travel, whatever it is, put them up in a hotel, take them out to dinner, and then they do their talk, and usually they get an honorarium, $100, right? For talking for an hour, that's pretty good, right? And you know, you brought them here, you do all of these things, right? That seems pretty reasonable. There are some instances where those honoraria get bumped up a little bit, but usually that's what you kind of are thinking, right? And maybe getting somebody here and all of that stuff might be about $1,000 when it's all said and done. You pull them in, you put them up in a hotel, you took them out to dinner. Under the table, you bought them some drinks because this is a dry campus, right? So all of that stuff, you kind of expect maybe about $1,000, right? When it's all said and done. So microbiology club is thinking, geez, wouldn't it be cool if we got Terry Mullis here? He can talk about stuff, talk about the discovery of polymerase, how he does all of this stuff, making these machines, right? Every time one of these things gets used, and they're used probably a dozen times in the biology building in a single day, he gets royalties off of all of this stuff, probably a penny, but he's still getting royalties, right? Also, good reason to kind of just retire him, right? Just get kickbacks. So we get a hold of him, not really, it's like movie star type of stuff. Our people talk to his people, right? Because you don't just talk to him on the phone. He doesn't have time for that, right? So we ask him, right? Would he be interested? Of course he would. His honorarium, 50 grand. And we're like, what? We don't have that kind of money, right? We're just a poor little microbiology club. We could, scrape together a couple grand, right, to get you here. We'd maybe give you $300, right? We said, no, thank you, right? Thanks for letting our people talk to your secretary's secretary, right? Is really kind of what happened, right? And done deal, right? And then I've heard other stories, like I said, he's kind of a little bit out there. Maybe it was for the best, right? We don't need that. So kind of crazy, right? All ultimately starting in Yellowstone as we think about this. And there are all sorts of advances that truly come about once Terry Mullis understands this heat tolerant polymerase, right? And as I said, the thermocyclers have definitely shrunk over the course of time. And there are some that are very small. The military uses some of these. You can throw them out of the back of an airplane or a helicopter dropped into the middle of a war zone. You can open this box up and you can do real-time or rapid PCR that can sample stuff like air to know if there's some sort of biowarfare agent that has been released, or there's some sort of crazy pathogen that we're dealing with. It can detect this and amplify it right before your eyes without needing to do microbiology and grow up the culture, grow up the bacterium, and add it to this machine, which is what we think about with most PCR reactions. We've extracted the DNA and we've done work with it behind the scenes to get it ready to go into this machine. There are other ones that basically do it all right there, right? Kind of crazy, right? And there are all sorts of other potentials that employ similar sorts of techniques and technologies into this mix. And we'll talk a little bit about that as we progress through. But you could take something that you know, this type of bacterium, and you can run it through a special type of PCR reaction, a sequencer, that's going to put in specific nucleotides that are either fluorescently or radioactively tagged, that as these sequences are being generated, they get detected, and you know the exact assortment or arrangement of the nucleotides, you know the genetic code, you know what this information actually says. You may not know what it means, but you know all of the nucleotides. And that's pretty impressive as well, all right? So how this happens, and you guys, I think maybe you extract some DNA. Are you extracting your DNA, or did you extract DNA? But are you going to? Because of COVID, probably stinking can't play with your own spit. Come on. All right, you can, if you are truly interested in something like this, there are some pretty interesting YouTube videos. I think Nova had one. I think that's the one we put into the lab to try to get you to do some of this stuff. A little bit of alcohol, isopropyl alcohol, some detergent, like, you know, dishwashing detergent, some of that, and some water, some salt water is really all you need, and a couple of drops of blue food coloring. And you can, over the course of just a few minutes, get some of your DNA, and maybe around a toothpick or a glass rod, as we've got up there, you can see the DNA within your mouth, right? Most of it, your stuff, but some of it's also some bacterial DNA as well. A lot of it's your own, right? So in order to get it, the cells get lysed with some sorts of detergent that disrupts the membrane. There are other things, and here we're not talking about just extracting it from your spit, right? We are trying to get DNA and only DNA. So here we can take the lysed solution and treat it with a couple of other enzymes. Proteases are enzymes that destroy proteins. RNase is an enzyme that destroys RNA molecules. Once you've treated this suspension with both of these enzymes, what you're left with is DNA, right? So you can put it into a centrifuge that spins it very rapidly. The debris, all of the junk gets pelleted down, and the supernatant, the fluid, has the DNA suspended within it. You can then collect that out, as we see here, with some alcohol. It will precipitate, and then as this happens, you get kind of, it looks to me, sort of spaghetti noodle-like as it twists around. When you do this, if you're doing your own DNA, it kind of looks like very, very small white strands. And as you keep twisting, you can get kind of a little glob or a goober of DNA, is sort of how it looks. It is kind of impressive. You're like, that's my own stuff, right? So once we have done this and extracted some DNA, now we've got the ability to actually do something with it, right? And it depends on what we might be thinking. Oftentimes, these strands of DNA get processed or chopped into fragments with other types of enzymes called restriction endonucleases. I like to think of them like scissors, molecular scissors, all right? They cut DNA in specific regions, specific nucleotides. It's not just randomly chopping stuff up into little bits, all right? A specific arrangement of nucleotides, and this enzyme cuts it, right? And there are handfuls of different enzymes, right? All of these, lots of these guys coming from bacteria that are breaking down nucleotides as they come across them in the environment and recycling those as nutrients or nucleotides for the bacterium to grow, right? But when we recognize these enzymes and what they did, again, like molecular scissors, it makes me think of people that scrapbook, right? I am not a scrapbooker. I am not married to a scrapbooker, thankfully, I think, right? Maybe not. Maybe you're a scrapbooker and you're like, don't say that, right? I think this because I saw a student that was a scrapbooker, and she was wheeling this big box of crap that I thought she's lugging all of her textbooks around with her. What in the hell? Why would you do that? Oh no, it's her scrapbooking stuff. And then my next question is, well, why in the hell would you be bringing that to class, right? That seems, because she's addicted, right? And in this box of stuff are multiple drawers. Are you scrapbookers?
No.
Okay, maybe. You're crafty, okay. So you're not bringing it to class. Maybe you will know what I'm saying though. In this box of stuff, she has got probably 20 different pairs of scissors, right? Scrapbooker. Lots of scissors, right? To make all sorts of crazy cuts. I had no clue, right? I was just like, holy crap, let me see those scissors, right? And now I'm like, maybe I'm a scrapbooker, right? I am not, right? But I was impressed with all of these scissors. And then that makes me think of this, right? If you've got a drawer full of 20 different types of scissors that make different types of cuts, that's the same as this drawer full of restriction endonucleases, this drawer full of DNA cutters. It all cuts different types of fancy cuts, right? Little bases, 5, 6 bases long that all say one specific thing. These scissors cut it. Next one that's got 7 different types of bases, another pair of scissors cuts that. So now you've got all of these possible scissors to cut your DNA in hundreds of different ways, and you can start making cuts with one type of scissors, right? I get this one pair of scissors and I cut this DNA, and then I'm going to run it out on a gel. I'll show you a picture of that in just a second. And all of the fragments, all of the pieces, as you've went to town cutting stuff up, you've got some things that are bigger, some things that are smaller, and it all looks this one way. It's a picture. These scissors made these cuts, right? And you know the cuts they make are a specific sequence. So now you got some hints about the type of DNA, what you're looking at. And then you take that, you take a picture of it, you set it off to the side, and you get your other scissors and you express those on the same chunks of DNA or the same bit of DNA, and it's different types of cuts. And when they run out on the gel, the picture is different. This is called a molecular fingerprint, right? When we think about something like DNA fingerprinting, we're thinking about like maybe a paternity test, right? There could be a handful of people that are possibly the father of this child. You would hope that maybe it's not like that, but it could definitely be that way. And in order to identify, you could take each of these individuals' DNA. You're also taking the offspring and the mother. You're getting all of this into the mix because the child's going to have half mom, half dad. You need to see what's going on, right? And in turn, express it with these scissors, run it out, and you can see things that, if you've got the father in the mix, are going to pair up and show the similar sorts of cutting patterns as that child, right? Crazy stuff, right? And it's all different types of scissors, right, that can do that. So that is what we're seeing right here, right? DNA fragments chopped up by a specific type of nuclease that allows for the DNA to be cut again, specifically, all right? Not just randomly, straight scissors, chopping stuff up however you want, all right? So we've got a couple on the sides that kind of frame it in. These are molecular ladders. So this is a known bit of DNA that gets put into the flanking wells that give you banding patterns of known sizes, right? And you can look at this, and based off of this gel, the DNA all goes in up at the top, right? And we can see a little of it lighting up here on both sides. It kind of depends on the ladders you're using. Lots of them now are already pre-stained. They've already got something in there, so as the gel is moving, you can see stuff migrating through. You don't see your own sample, but you can see other stuff. And as you know, looking at this ladder and the package insert shows you a picture, this is how it should look. And there's maybe, a dozen bands. You start watching them show up, and once they look very similar to what the package shows they should be like, your gel has ran for the proper amount of time. Usually it goes about an hour as you're running this stuff out. So as we see on this picture, larger stuff toward the top and smaller stuff toward the bottom. This is a gel. It's like Jell-O jigglers, right? This is something that you could hold. You probably aren't going to be. You could feel it. It is Jell-O-like, all right? With that said, it's not just a solid mass, right? It's kind of like a sieve, like a strainer, right? You dump your spaghetti noodles into this strainer and the noodles stay in the strainer, but the water goes through, right? This gel, once the DNA is put into these wells, and there's little teeny slits that you pipette small amounts of the DNA into, you express this, you put it in a chamber and put some electricity into it, and they start to migrate. Bigger stuff going through that colander, right? Bigger stuff gets stuck, doesn't move as rapidly, doesn't move as far into the gel, and littler things move pretty quickly, right? So it separates based off of size. Smaller toward the bottom, bigger at the top, and if you're not careful when you're running these gels, you can shoot the small guys right off of the gel into the buffer, and then you've lost part of your sample, right? So usually you've got a good chunk of gel at the bottom that is kind of just a zone that you don't want to go into because you might be losing some stuff, right? So we can see here, there were 6 different bits that we're looking at, 6 samples run on a gel. It looks like more than 6 to me. As I'm looking at this, right? That looks like 7, whatever. There are different patterns, right? Some similarities and some different, all right? And each of these individuals, if we're talking different people, all right, have some variation in these banding patterns, all right? This in turn lends some light on the specific samples. And like I said, you could do this with one type of scissors, and then you get your other scissors out and express that to the DNA sequences and run them out on a gel and see a completely different banding pattern. And you can do that a handful of times. And each of these bands is a different piece of DNA, smaller ones, or clear up here, great big pieces, right? That you have cut out, right? And you could definitely do some crafting with that, right? You've got all sorts of different pieces, right? That's pretty interesting, right? So maybe as we have chopped this out, maybe some of these specific things are a gene. Maybe this is a gene of interest. There's, maybe 500-600 different nucleotides that makes up this fragment right here. And maybe that's something we want to do more science with, right? We can take this band and we can extract it out of the gel, and we can take that DNA and put it into that thermocycler and amplify. Now we've got 1 billion copies of this one band, all right? Now we can do something with it, right? because instead of just having one little teeny piece, now you got a whole pile of it, all right? And that you can definitely do some science with. Or crafting. All right, so the PCR reaction, it is a chain reaction. It happens very rapidly in this thermocycler. Usually in the course of 3 to 4 hours, the cycle is complete, all right? Lots of times when this happens, researchers that do a lot of PCR reactions, they will spend a lot of the time the day getting their samples ready, prepping them, dealing with stuff, chopping them up with nucleases, doing whatever they've got to do. And then as we move toward late afternoon, we start setting this up, right? The thermocycler, it can do all of this stuff on its own, right? You don't need to be there and babysit this machine. You can put your samples into it. And as soon as you put them all in and you type in your respective parameters, because you can do things a little bit, you can tweak them a little bit. As we see here, different temperatures, you may adjust it the littlest bit, right? Once this thing has ran for 3 or 4 hours, that machine can just go into standby mode. And the thermocycler that was acting like a heater, cooking things at various temperatures for a few seconds, now turns into a refrigerator, and it holds everything at 4 degrees Celsius, keeps everything stable, just like you taking the samples out and put them in the fridge so you could deal with them later. You could set this machine up at 4:55, you push go, you walk out the door, you go home, the next morning you show up, open the box up, everything's chilling in the fridge, and you're good to go. You've got all of your samples, right? Pretty crazy, right? So we start with a chunk of DNA, right? A fragment, like one of those that we've maybe pulled out of that gel, right? Not the entire chromosome, a hunk of it, right? So we've got the double-stranded DNA, again, base paired by hydrogen bonding, right? And we're going to break those apart by denaturing them at 95 degrees Celsius, almost boiling, right? Super hot, it causes the DNA to fall apart. And now we've got 2 separate strands. And remember, when we're copying DNA, the 2 parental strands serve as templates for the new daughter strands that are growing down below. So we need polymerases. We also usually need to know a little bit about the DNA that we're trying to replicate in the first place. You've probably got some hints if you used these enzymes to chop it up in the first place. You know a little bit about your scissors and what the DNA looks like, at least at the cut sites, right? This is where my scissors made the cuts, right? I know these nucleotides must be these 3 bases or 7 bases or whatever it might be, right? You develop some primers, and the primers are RNA primers, right? RNA primers are what DNA polymerase needs to link up to this growing strand of DNA. That happens in our own bodies, right? It's RNA primers for this to occur, all right? So the RNA primers link up or anneal, they base pair, and this provides, the little yellow guy right here is the DNA polymerase. It provides the docking area for polymerase to link up to the sequence, right? So in our test tube of material, we've got our strand of DNA that we want copied. We've got some primers that are going to get used. We've got a bunch of nucleotides and our polymerase, right? All of this stuff together, we call the master mix. It's all the ingredients, right? And once it's done its job, you've got your product hours later, all right? So we start off by heating at 95. It causes the double-stranded DNA to break. Now we've got the 2 templates, the single-stranded DNA. The temperature goes to 95 for seconds, right? It only takes a few seconds for the DNA to get denatured. As soon as that happens, it drops it down to 50 degrees Celsius, cools it very rapidly. At that temperature, the primers are able to link up to the DNA. At that point, after another handful of seconds, right, it's very rapid. After 10 seconds for primer annealing, the DNA polymerase is able to link up as the temperature gets bumped up to about 72. And again, these can alter the littlest bit. At 72 degrees Celsius, polymerase does its action. It moves along both strands of the DNA molecule. We're not going to go through details with that, but it does it very rapidly. Again, a handful of seconds, and you've made this copy, right? Once this has happened, it's basically a repeat, right? As soon as this occurs and we've got our copied strand, the temperature bumps back up to 95. And now instead of separating one, we separate two. Those two generate four. Those four generate eight, right? And then 16. It's exponential growth. All of the sudden, you've got 1 million copies that get separated, and it doesn't take very long before there's billions of copies, right? Handful of hours, right? And we can have massive amounts. Just like that picture with the test tube and the DNA around that glass rod, your master mix starts off as a colorless liquid. It goes into the thermocycler, and 4 hours later, there is a glob of DNA in that tube, right? Because we've replicated it countless times, right? Giving you huge amounts of DNA, giving you the ability to actually do something with it, all right? One piece isn't enough, but 1 billion copies, now we're talking business, all right? And there's all sorts of things that we might want to do. You could also, flipping it back to this, have these specific sequences that we've chalked out. And now we could flip it to some bacteria and start thinking about, well, what do these things do? And you could take the respective genes and amplify them, as we've just shown, and then take those respective products and put each of them into a bacteria and see what that bacteria is able to synthesize. We call that a clone library, right? It's a library of all of the individual pieces or fragments put into something, and then you can see what the protein expression is, right? What is it that it's building, if anything? And usually it's building something. So here we can see those scissors, right? We've got our sequence of DNA. This restriction enzyme, the red bit, are the scissors, right? And so it sees a sequence of nucleotides, all right, read a specific way, all right, and it's read the same 5 to 3 as it does the opposite, right? So it's a repeat, a tandem repeat, right? So mirror image, CCTAGG, GGATCC, right? Palindrome is what it is. And with that, it makes the cuts. And in this case, in between the G's, what you're left with are these fragments. And these are the two parental strands. And this is the new stuff lining up. Most of the time, these enzymes make cuts that are jagged, right? We call them sticky ends, right? Because you got bases on either side, right? There are also scissors that are just like regular scissors that most of us probably have at our house, not the crazy scrapbooking scissors, just a regular straight cut. When they cuts the DNA just straight across, we call those blunt ends. And that is very difficult to do molecular biology with. You can do it, but it's a little more difficult. It's a lot easier to get a chunk that's got these little overhangs to stick into a new chunk of chromosome. As long as you cut them with the same scissors, they'll pair up. If you cut it with just a straight scissor, it's a little more work. And the potential for that to get flip-flopped around and now be in the reverse order is considerably greater than if you've got the sticky ends that make it so they have to pair up this way, right? That helps to ensure the sequences are going in the right orientation, right? So it makes the cuts, you've got these ends, and then we can start replicating if we're talking PCR stuff. That's not what we're talking about right here. This is just the cut, right? This is what's left over, right? And if you just quickly cut these and give them a second side by side, they'll just stick back together, right? So there's an overwhelming amount of enzyme that makes multiple cuts to ensure you don't get a lot of it sticking back together. So I talked about the clone library. That's really what we're looking at right here, all right? So let me flip it back a couple. As we look at this, we've got a handful of different things that we can see. If we want to take each of these that we've cut with these scissors, and maybe these same scissors making these same cuts, give me 4 or 5 different fragments. Each of those fragments can be put into a chromosome. In this case, almost always, it's a plasmid, which is extra chromosomal material that bacteria can have. The bacteria doesn't need the plasmid, but if it has it, it usually gives it some advantage, right? The plasmid is something that's easy to manipulate. If you try manipulating the bacterial chromosome itself, you possibly destroy things that the bacteria needs for survival. That becomes an issue, right? But if you manipulate the plasmid, it doesn't do anything to the bacteria's chromosome. It still lives. And it also synthesizes whatever that plasmid now says, make this too, right? Make some human insulin while you're just growing in this big vat, right? Do some of that for us, right? So there's a lot of possibilities, and there are lots of things that we are potentially interested in that we could incorporate into these plasmids and then express them. And that is a clone library, or this molecular cloning, right? It's not the same as the Dolly the sheep that we started this unit of stuff with, although you're going to see her in just a second, right? And we'll talk about that type of cloning in a little more detail. So here we've got the orange stuff is the foreign DNA, the stuff that we're interested in. Our plasmid over here, it's got a restriction site that the scissors will cut, and we use those same scissors to cut the foreign DNA. So now we've got sticky ends that match the sticky ends of the plasmid, and that allows for these fragments to get incorporated in there, right? Now we got a couple of different plasmids. Each of these orange bits could be larger or smaller segments, but the sticky ends are the same, right? So if we imagine these are 3 different genes from this one chunk of DNA, we can now see what these 3 genes do as we take these plasmids and we put them back into the bacteria that they came from in the first place, right? So the plasmids get back in there. In this case, we can see some of them maybe don't get it, right? You can grow these up on a plate, a petri dish, and oftentimes with this type of stuff, most of the plasmids can, impart some sorts of advantages to the bacteria. A lot of them are things like antibiotic resistance. So now you start thinking about some sort of super pathogen, right? These super bugs that are resistant to all sorts of things. Well, this isn't something we're thinking about human disease. This is something we're using in a lab. So this plasmid says, oh, you're resistant to ampicillin, right? Well, if we put ampicillin into the medium, if all of this stuff functions like it should, then the gene to be resistant to ampicillin is there. It gets turned on. They grow up in the presence of ampicillin and they grow. That tells us the plasmid got into the bacteria and it's doing what it should. If they don't grow, the other guys, it's this, right? The plasmid didn't get in there, or it didn't function the way it should have, and they're not resistant to the antibiotic, and it kills them. So only the ones that are resistant are able to grow. We pick them off knowing that they are clones, that they are mutants, if you will, because we've altered the sequence of DNA, right? And now each of these can be picked off. So this is a colony of bacteria, and you can plate it out onto a new plate to grow more bacteria. And now you can have specific ones with their respective genes in there that you're growing in mass quantities, right? And expressing those specific proteins, whatever they might be. So this is cloning, but at a larger scale. When we think about, the start of this, we showed Dolly, but we didn't go into the details. I told you a little bit about it, but we didn't see any of this, right? So here we have the two sheep. So we've got a Scottish blackface. This was the cytoplasmic donor. And the Finn Dorset, the white-faced sheep, this is the nuclear donor, all right? So this one donates the egg. This one donates the diploid cells, all right? So through enucleation with a very small glass pipette and a dissecting microscope, the egg has the nucleus sucked out of it. So now we've got just the hollow shell of the egg with no genetic information in it, right? We take the mammary cell, again, diploid cell, and the shell of the egg. We cause them to fuse with a jolt of electricity. The current pulse stimulates these cells to join. And now you've got a diploid cell that is within this egg, right? And that jolt of electricity causes it to start the cell cycle, and it starts dividing. It basically is a stem cell at this point, if you will. This becomes the fertilized egg, but it is the nucleus of this diploid sheep, and it starts replicating. Going through multiple rounds of division, this fertilized egg, if you will, becomes this blastocyst, a handful of cells. They keep replicating. We see, the embryo developing within our surrogate sheep. The one that donated the egg is the one that gave birth to Dolly. And Dolly looks just like the Finn Dorset because that's the nuclear material that said, this is me, right? Which is her. And Dolly, as I mentioned last time, when she was born, she was already middle-aged because the sheep that donated the mammary cells was already middle-aged. Her DNA had already gone through multiple rounds of divisions. It had already gone through this process countless times, and here she shows up, right? So, looking nothing like the mother that gave birth to her, right? Because none of her information is in this sheep, right? It's just the cell, the shell of that egg, right? No nuclear material. It's all coming from this one, right? And it took numerous attempts to get this to function, just like, with different fertility situations, oftentimes people that are struggling to have a child, it's not the first time they go visit a doctor that they end up pumping out a kid, right? It typically takes multiple visits, right, with that. And it's a similar sort of an idea like this, multiple times before it actually took, if you will, right? And the egg was able to implant as it should and become Dolly that we see hatching out. However many months later it takes for a sheep to have its little lamb. I don't know the answer to that. So pretty interesting, right? And it sets the stage as we look at this, as we start thinking about mammals being cloned. We can go back to this and bacteria. We've been doing this type of cloning for a long time. There's no issues with that, at least none that I really have heard about as far as concerns. But we look at this and it raises some flags. There are some potential issues. If you could do this Why could you not clone a human? There's really no reason you couldn't, right? There are numerous reasons why you probably shouldn't, right? And in turn, one of them specifically is the fact that your little clone of you would already be middle-aged or whatever, the age of those cells that you took it out of, right? And that sets the stage for problems, right? Should you be cloning yourselves? It's hard to say.
So it was like
I can't remember how long Dolly lived. A couple of years, a year or two, something like that. Not very long, because she had already lived, if you will. Her DNA basically, the telomeres at the ends of all of our linear chromosomes, every time that DNA replicates, a little bit of that telomeric DNA breaks off. It's really the aging process. And so if you've gone through, 30 rounds of division, and in sheep that, live for maybe a decade, I don't know how long, I don't know the longevity of a sheep, but I'm assuming it's probably about like that. In those circumstances, that DNA has already replicated a bunch of times. And so those telomeres are already half the length that they should have been if it was really a brand new sheep. So as far as the full, the actual amount of time that Dolly survived, I'm not 100% with that. But I do know she died of old age at a very young age, which seems weird, right? Oh, someone's chatting for me. Somebody's got details up in here. 6.5 years, it says on Google. Is that 6.5 years that Dolly lived? I assume probably. Yes. Okay. And so, Amanda, while you're on there, how long does a sheep live normally? That's what we want to know now. Come on, Google.
11 to 12, I think.
11 or 12. All right. So she lived
I
about
just Googled it.
Okay. Thank you. Okay. So interesting, right? And it's, I also think this is interesting. Just also showing that surrogate was just a surrogate. All she did was carry this. She had no genetic input into Dolly in the first place, which is pretty impressive. So this, as we think about cloning, as we think about all sorts of other ways to manipulate things, there are lots of other questions, not just, oh, should I clone myself, right? There are ideas about all sorts of genetic problems, genetic issues, anomalies, diseases that have a genetic basis. Knowing the specific sequence of DNA and knowing the issue, the error, or the mutation that has lent to that problem begs the question, could you not fix that? Could you not correct those problems and in turn eliminate those issues in the first place? Seems like a brilliant idea, right? And the answer is yes and no, right? There are definitely some opportunities where we have cured some diseases, right? We have also caused some big issues, right? So at least right now, we're not thinking about curing. We think about treating. That's more where we're at with this. And it's a lot easier to try to treat specific things than to try to cure, all right? So we'll think a little bit about some of these things, right? There are definitely some genetic defects that we deep down really would say it would sure be nice if we could cure that, right? If we could eliminate that in the first place, that would be fantastic, right? Another story for you. This is back in the 1970s. There was a boy that was born with a genetic disease called SCID. which stands for Severe Combined Immunodeficiency Syndrome. It's kind of like AIDS, but it's something you're born with, not something that you acquire from a virus, right? So basically, he's born really without an immune response. He's got no immunity. He's got no capabilities of fighting off anything, all right? So right after he's born, his parents know that there are issues, and basically, he's not going to survive, all right? So there's all sorts of counseling and details and all sorts of concerns. He does survive. He survives in a sterile environment. They put him in a bubble, if you will. His David the Bubble Boy. There's a movie about him, all right? David lived in this bubble. He lived in this sterile environment till he was 12, right? That's a super long time to be living in a bubble, all right? There are pictures. I don't think there's pictures in here. There should be. Oh, those mice, cute little guys. This big chamber where he lives, there are these gloves where people could put their hands into the gloves. They could give him a hug. They couldn't hold him. They couldn't touch him. They couldn't actually interact with him because simply that contact is going to be a death sentence, right? So anything going into the bubble is sterilized. All of his food, nothing like you think about a little kid eating all sorts of crazy things. No, or maybe a little kid not eating all sorts of crazy things. That's more like him, right? A very bland and sterile sort of situation, right? Not a good way to be a kid, right? And most kids want some contact. They want a hug. They want some nurturing, right? Most kids do, all right? So the thought was, you can survive in this circumstance and we're going to find a cure for you. We're going to fix this, right? We're going to figure out a way, right? And there was lots of promise. There was lots of hope that something was going to happen, right? And after a little over a decade of living in this seclusion, lots of counseling, I'm sure, he decided that this is too much. He's done. He doesn't want to live like this. It's not a life in the first place, right? So he basically crawls out of this bubble, embraces his parents, and he gets sick and he dies, right? A pretty sad situation, right? Not very long after David dies, they cured SCID, right? It's not cured, period, but there was an individual that they detected was going to have this, and they did a process kind of like what we're showing up here, where they target the specific stem cells and they correct by putting the right gene in, and then it's in a stem cell, it becomes part of this child and it's corrected, all right? So this child's born and it doesn't have SCID. And there's another one as well. I think it's Wiskott-Aldrich that we also have seen some cures, right? Where we've gone in and corrected stuff. And in that situation, life expectancy is very low. You don't make it to puberty in those circumstances. So another one, cured, all right? A couple of genetic diseases that we showed we can do this, all right? And then there's a couple of these kids that got treatment, and after they were born, very rapidly, they developed some crazy cancers and they died. And they died because the genes that we were trying to put into them to correct these problems didn't go into the right cells or didn't go into the right cells with the proper proportions. And it caused all sorts of issues and death. And in medicine, that's kind of a no-no. We're not here to hurt you. We're here to help you, right? That's the idea. And so once they recognized these problems, they kind of put the brakes on and said, you know what? curing is something we shouldn't be thinking about, right? We should think about trying to treat instead of cure, and hopefully, over the course of a little more time, we will get some of this stuff figured out and be able to specifically target, to be able to find the proper proportions and not cause adverse events, right? So David ultimately died, was unable to be helped. There were a couple that have been cured. And now we start thinking more, like I said, the treatment. You can take specific cells, like thinking bone marrow transplant sort of an idea. You can take some cells out of the individual and you can manipulate these cells in the test tube, right? You're not manipulating their specific stem cell. You're not manipulating that that's going to be part of them, period. It's the cells that have already been generated, and you fix them. And then you put them back into the individual. This is the therapy. You're treating it. You're not curing it, right? Most of your white cells, about half a year or so that they can persist. Your red blood cells, about 4 months or so that they persist. So these cells, we know there's a specific kind of rate of turnover, and individuals that we are treating with some of this therapy situation requires that they're kind of tied to a clinic where they go in and get blood drawn off, manipulated, and put back in at least twice, maybe 3 times a year. This has to happen, but these people don't need to live in a bubble. They don't need to be concerned about, there's still some concern, I'm sure, but it's not the same because they're functioning with cells that are actually doing what they should be doing, right? It's pretty impressive. It's pretty fascinating to think about that. A pretty newly discovered group of proteins and enzymes in some bacteria is lending to some other opportunities. I think we're on the cusp of some pretty fascinating stuff, and the ability to alter and manipulate things is right at our fingertips, right? The CRISPR-Cas mechanism is really kind of like bacterial immune system is kind of what it's like. And it helps that bacterium to deal with things coming in, like maybe virus, and it helps to ensure that if that virus is trying to get into the genome, it chops it out. Well, now we've recognized that this battery of things we can use, like those molecular scissors, like these restriction endonucleases, to chop, but we can use it differently. And not only are we cutting things out, but we can use it to be implanting or putting things in as well. It sets the stage for manipulating stuff, like putting that gene into a specific cell in the proper orientation. The stuff that I said, wait a minute, as they put the brakes on, we don't know what we're doing. Now we think maybe we have a way to do that, right? The potential for designer babies is coming up, all right, which I think might be a little bit disturbing, right? There are definitely possibilities where you may be concerned about what your child could potentially be born with. Karyotypes may lend some ideas into that, but then it also begs the question, do you want to have some specific trait? Do you want some characteristic in this child that's not born yet? Do you want to try to decide what that child truly should be? I kind of wonder if that's something you should have no business in doing in the first place, right? But I think we're marching toward that. Okay, guys, we will stop with that. Questions or concerns? Farewell. Thank you for looking up Dolly the sheep, Amanda.
You're welcome. Thank you.
Have a good day.
You too.